Essay. A cycle is a biological pathway or process in which the end product from one cycle becomes a starting point for the next. Write an essay about cycles in biology. Biological cycles involve the recycling of some elements. An example in which there is regeneration of molecules is the Calvin cycle in biology in plants. The Calvin cycle takes place in the stroma of the chloroplast of photosynthetic plants. The enzyme Rubisco converts ribulose bisphosphate into glycerate 3-phosphate when carbon dioxide is fixed. Initially, the 5-carbon compound is made into an unstable 6-carbon compound, which then dissociates to form two 3-carbon compounds. These glycerate 3-phosphates then react with the hydrogen supplied by the reduced NADP from the light-dependent reaction, formed in non-cyclic phosphorylation, and with the help of ATP from the light-dependent reaction, triose phosphate is made. The triose phosphate can then, every 6 turns of the Calvin cycle, be converted into a 6-carbon compound, hexose sugar, and then the remaining 5-carbon compound is converted back into ribulose bisphosphate, and thus the cycle completes. The importance of this cycle is the generation of hexose sugars, which can then be made into many other compounds needed by the plant for growth and repair, for example cellulose, and this is done using light energy from the sunlight and carbon dioxide in the atmosphere. The generation of glucose as part of hexose sugars is something that can be stored as starch in plants for use at a later date in order to generate ATP. This is done in aerobic respiration, which includes the Krebs cycle. In the Krebs cycle, the oxygen that diffuses into the matrix from the rest of the cell is used in oxidative phosphorylation. The pyruvate that is generated from the link reaction enters the mitochondrion. The Krebs cycle starts with the 4-carbon compound oxaloacetate, and this combines with the acetate from the link reaction, which is passed into the Krebs cycle by acetyl coenzyme A. When the two compounds combine, the coenzyme A returns to be recycled. The new 6-carbon compound formed is called citrate. The citrate is then altered and releases carbon dioxide. The hydrogens that are available are used to reduce NADP to NADPH. The hydrogens that are available are used to reduce NAD to NADH in respiration. The cycle continues with a 5-carbon compound that is further decarboxylated and oxidized, losing 1 carbon dioxide and a number of hydrogens which reduce NAD and FAD to NADH and FADH. The change in the substrates from one type of 4-carbon compound to another type of 4-carbon compound releases energy which is used to form ATP from ADP and inorganic phosphate by something called substrate-level phosphorylation. The cycle continues once the oxaloacetate has been made. The formation of ATP is also made when the NADH enters oxidative phosphorylation, and as it loses the hydrogen ions to the system, the recycling of NAD to return to the Krebs cycle, then the electrons pass down the electron transport chain and the hydrogen ions are pumped across the membrane. This sets up a proton motive force, and the hydrogen ions flow back down a concentration gradient in a way that allows the enzyme ATP synthase to generate ATP, recycling the ADP and phosphate made when ATP is hydrolyzed to release energy. Energy is required to be passed through the food chains in the form of biomass. This is the carbon cycle. In that, sunlight energy allows carbon dioxide to be fixed as part of photosynthesis. This then leads to the formation of carbohydrates in the plant species, which are then part of the carbon cycle. Compounds of carbon are passed between each trophic level by the consumption of the organism in the previous trophic level. The ingestion of starch by cows can then be hydrolyzed into glucose using commensal bacteria in the gut of the cow. The glucose can then be absorbed into the cow's bloodstream and transported to the muscle cells, where it is taken up and converted to glycogen and stored. When the cow is eaten in the next trophic level, the glycogen is taken in and digested and broken down into maltose and then glucose, then absorbed and assimilated. The same happens to the protein compounds in the plant. These are digested in the cow and the amino acids released and absorbed into the bloodstream and then converted by cells into proteins, such as those found in hair of the animal. If the animal molts, the proteins in the hair can be broken down and carbon compounds enter the soil. Decomposers such as saprobionts can then hydrolyze the large compounds into smaller compounds and use carbohydrates as a source of energy. The carbon dioxide released by the saprobionts and all other organisms in the carbon cycle as part of respiration can then be released into the atmosphere, and these compounds are then absorbed by diffusion into the plant leaves, and the plant fixes the carbon dioxide into carbohydrates by photosynthesis. The cycle continues with photosynthesis taking up the carbon dioxide and respiration releasing it into the atmosphere. As part of the carbon cycle, other nutrients are released when the animals molt or urinate or defecate. Nitrates in these compounds from dead plants, dead animals enter the soil and ammonification takes place by saprobionts. The ammonia compounds are then converted to nitrites and nitrates by nitrification. The bacteria that carry out these reactions allow the plants to take up nitrates, which then incorporate these nitrates into amino acids, which can then be taken up into animals by ingestion, and the animals carry out digestion and break down the amino acids in the proteins, and then these amino acids are then assimilated into the proteins of the animal and become the animal's biomass, and these will be released into the soil when the animal dies, or passed on to the next trophic level if the animal is consumed. The nitrates in the soil may then be recycled into nitrogen by denitrifying bacteria, so the nitrogen cycle involves the uptake of nitrates from the soil and then into the plant, through the trophic levels, back into the soil through urination, or into the soil by decomposition. The nitrates assimilated into the muscle include cardiac muscle. The cardiac muscle is important for the pumping of blood. This system is called the closed system and a double circulatory system. The blood leaves the left ventricle and into the aorta, then around the body and through the organs. The blood then returns to the heart via the vena cava, then from the vena cava into the right atrium, down a pressure gradient into the right ventricle. The cardiac cycle then continues when the muscle contracts, reducing the volume, increasing the pressure, and blood moves down a pressure gradient and into the pulmonary artery. The blood then enters the lungs and back into the left side of the heart as oxygenated blood via the pulmonary vein. The blood then enters the left atrium, down a pressure gradient into the left ventricle, and then leaves the heart via the aorta, down a pressure gradient. The cardiac cycle allows oxygen and glucose to be delivered to the tissues for aerobic respiration. The cells require respiration for ATP for active processes, including cell division. The cell cycle involves cell division in the form of mitosis. This involves prophase, metaphase, anaphase, and telophase. This is followed by cytokinesis, the division of the cytoplasm, and then growth phase when one organelle replicates and generates ATP, and then by semi-conservative replication the DNA of the cell is replicated and copied, and this is called the synthesis phase. Following synthesis phase there is growth phase 2, when the cytoplasm grows and the organelles get involved in mitosis, get divided and copied. The cycle begins again with mitosis and nuclear division. Cells divide by mitosis for growth and repair of the tissue. Damaged tissues in need of repair include muscle tissue. The muscle contraction also shows a cycle. In muscle contraction, the ratchet mechanism involves the use of ATP. Initially, the calcium released from the sarcoplasmic reticulum binds to troponin, which moves the tropomyosin out of the way of the binding site found on the actin molecule. ATP binds to the myosin head, and this allows the myosin head to detach from the actin molecule. The ATP is then hydrolyzed into ADP and inorganic phosphate, and the energy release causes the head of the myosin to change position into a cock position. The myosin head then binds to the actin binding site as phosphate is released. Then ADP is released, and this allows the head of myosin to return to a lower state, flicking the actin filament. Then the cycle begins again, and the ATP binds to the myosin head, releasing the myosin head from the binding site and allowing the actin to be moved closer and closer together, causing muscle contraction, and this is cyclical.